All right, in this next video, we're going to talk a little bit about RNA, ribonucleic acid, which is another type of nucleic acid related to DNA. It's the next step in the story of going from DNA to traits or proteins. So how do the two nucleic acids compare? So you can see here in the picture, one of the differences is the sugar. DNA has deoxyribose sugar. RNA has ribose, which has one more oxygen than deoxyribose. RNA is generally single-stranded, not double-stranded, you can see in the picture here. And RNA contains the nitrogen base uracil in place of thymine, which is in DNA only. So there are three main kinds of RNA. The first kind is messenger RNA, or mRNA. An mRNA molecule is a copy of a portion of the DNA that will be used to make a protein. It's the copy of the gene from the DNA that makes a particular protein. So it's a segment of DNA gets copied, not all the DNA that gets copied to make messenger RNA. What messenger RNA gets made depends on what kind of cell it is and what the cell needs to do. So after being made in the nucleus, which is where the DNA stays, the messenger RNA is going to travel out of the nucleus through the nuclear pores to the ribosomes, which are in the cytoplasm. And remember, ribosomes are where proteins are made or where protein synthesis occurs. The next type of RNA is ribosomal RNA, which along with protein makes up the ribosomes. And there are two subunits, a large and a small subunit. Both prokaryotes and eukaryotes have ribosomes, but the structures are different in prokaryotes and eukaryotes. But ribosomal RNA made in the nucleus and then is assembled along with the proteins to make the ribosomes out in the cytoplasm. So, like I said, there are lots of different kinds of ribosomal RNA and there are lots of different kinds of proteins that make up the ribosome, at least 80 different proteins in the ribosome itself. Then transfer RNA, the job of transfer RNA or tRNA is to bring the amino acids of the building blocks of the proteins to the ribosome. So during the synthesis of proteins, you get the messenger RNA, which has the instructions, and then the transfer RNA brings the correct amino acids. So there's an area on the messenger RNA called the codon that codes for one amino acid or one building block of the protein. And on the corresponding messenger or the transfer RNA is the complementary copy or the anticodon to that codon. So how does RNA synthesis occur? This is called transcription. So we already learned about replication, which is the first part of the central dogma, where DNA gets copied. Now we're going from DNA to RNA, from one kind of nucleic acid to another, in transcription. So in transcription, a piece of the DNA is a template for making an mRNA copy. On transcription, we copied the entire DNA, and we used both strands as templates. In transcription, only one strand, the coding strand, is used to make the messenger RNA, which is a single strand molecule. So here you see RNA polymerase, the enzyme that copies the DNA message into RNA. Here's we call the transcription bubble, which is where the DNA is opened up. And again, one strand, the coding strand, is used to make the messenger RNA copy. And here's the growing messenger RNA being produced. So it all starts with promoters. So RNA polymerase reads the DNA sequence on the coding strand of the DNA, adds the complementary RNA nucleotides to the messenger RNA. But to start the whole process, the RNA polymerase has to bind to an area called the promoter. And so there's a promoter for each gene. So that's where the RNA polymerase starts the process of transcription. Now, once the RNA is made in a eukaryote, in prokaryotes, the messenger RNA is made just how it's supposed to be made, doesn't have to do anything else. But in the eukaryotes, there's going to be some editing of what we call the pre-messenger RNA or pre-mRNA. So there are sequences in the DNA that code for traits and things that don't. So the parts that code for the trait are called exons, and the intervening parts or the non-coding parts are called introns. So the cell has to cut out the introns, splice the exons together. So here you can see the introns, they're being cut out, and you get the mature messenger RNA produced, which just has the coding sequences, the exons. Also, a 5' cap and a poly A tail are added to protect the messenger RNA once it leaves the nucleus and goes into the cytoplasm, so it doesn't get broken down too quickly, so it has time to do its job to code for the making of a particular protein. So like I said, in eukaryotes, the genes have introns and exons. Prokaryotes, that's not the case. Introns are in the non-coding, exons are the coding sequences. Introns are cut out, exons are spliced together to make a mature messenger RNA. But they're not always spliced together the same way, and it makes the DNA efficient because a single gene can code for more than one protein. So depending on which exons are put together, determines which protein that gene produces.